follow up. I would like to, before I would start, I would like to give you some short introduction to about our agency, which is the Agency for Health and Food Safety. And we are in a good position to have kind of One Health approach, so we can have data we can analyze, monitor, assess, and research, and also communicate to in a One Health approach. And this enables us to give very make a good monitoring and show to the public, as we can do for West Nile virus. And West Nile virus is one of these very good examples which needs a One Health approach. And so what we can do, what we are doing currently is we have something that's called the Tierseuchen radar, which you can find using the QR code on the upper side of the slide. And there you can find data on West Nile virus, but also on other disease, which is monthly actualized. So, and the backbone of this data is the Aedes, the Animal Disease Information System. Furthermore, we have a website which is in the middle of the slide, where you can find the weekly actualized data of West Nile virus in human, vet, and vector data. Actually, the human data is the responsibility lies in the medical university, but I will come to this in a second. So, you can share or you can have a look at these informations on the website. But coming to the West Nile virus, which is starting with the tick, with the mosquito, sorry. It's the main host are birds. These are the reservoir, these are the amplifying hosts. So this is the cycle which is the one which is amplifying and for distribution. But humans and horses are kind of accidental hosts, which can also quit the West Nile virus. Looking at the birds, they are very, very often asymptomatic, but as I mentioned, these are the wildlife reservoir, and there are kind of several birds which are more susceptible, like passerine birds, and which means, and in this group, mainly the corvids, or also the birds of prey are birds which can develop very, very hard symptoms and also have a fatal outcome of the disease. We for the symptoms, it's normally that you have central nervous system symptoms, lethargy, coordination difficulties, ataxia. Some very often they have the head tilt, the head moving to one side, but also bleeding. The pica or the cloac can be found. The horses are also very often asymptomatic, but in a few cases, approximately in 10% of the cases, they also develop CNS symptoms like ataxia, depression, lethargy, or a dropping of the head. And which is really kind of terrible is that of these 10%, about 14 can have a lethal outcome for the horse. So it can be a really bad situation for the horse and the horse keeper. The human is mainly asymptomatic, but if there are symptoms, then it's more like flu-like symptoms, headaches, gastrointestinal symptoms. So, and the horses and the humans represent dead-end hosts. That means the transmission to the mosquito is not possible. For the West Nile virus itself, it belongs to the Flaviviridae, genus Autoflavivirus. It's worldwide, can be found worldwide, but except of the Antarctica. Actually, I think it's assumed to be the most widespread flavivirus. around the world. And until now, there are 9 lineages known, and lineage 1 and 2 are considered to be the most important. And lineage 1 is the one which can be found worldwide, mainly in USA, Australia, Middle East, sometimes in Europe. And lineage 2 is more distributed in Africa and is now draws responsible for the main outbreak in Europe, main outbreaks in Europe since 2004. In previous days, it was assumed that the lineage 1 is more pathogenic, but it turned out that both lineage 1 and 2 can make really bad outcomes for humans and be draw responsible for terrible fatal outcomes. Looking back in history, uh, the first finding was 1937 in blood of a woman from Uganda. Uh, Later on in US, you can find, it was found West Nile virus since 1992. I think I could make a whole presentation about the situation in US because there were some really bad outcomes. So this would most probably need another presentation. In Europe, just to give you some data, it was first found in 1962 in humans and equines. Later on in 1996, there was an outbreak in Romania with 17 fatal cases in humans. And 2003, 2004, it uh, occurred in Hungary with lineage 2. And late in the recent past, it was 2018, where Greece, Italy, and Romania mainly was affected with a lot of human cases, 1,500, and of these, 166 were fatal. And we also observed some diseased horses in these areas. 2022, another outbreak with approximately 1,340 people, 104 death cases. In Austria itself, we noticed the first clinic case. It was a bird of prey in 2008. 2009, a human case followed. And since 2011, we have some surveillance program on antibodies in horses. And maybe some interesting point is that it was also found in blood donors, in human blood donors in 2014. For diagnostic, it's not always easy to diagnose West Nile virus. Especially it depends which material you can get, which tissue, which kind of substrate you use. Mainly we use serum, urine, liquor, or organs. And for the animals, as I already mentioned, the Aedes is responsible. For the humans, it's the National Reference Center for Flavivirus at the Medical University. In Aedes, we use some kind of testing system using different ELISA approaches, different types, and it's possible to exclude and to consider several other disease and to come to a very good or to a very reliable diagnosis. So what we have to, well, what we are going to use is some flavivirus, pan-flavivirus ELISA, but you can also have to consider tick-borne encephalitis or pseudovirus, other encephalitis virus like the Japanese encephalitis virus or the Eastern equine encephalitis virus or the Western equine encephalitis virus. What you also have to consider is if there are IgM antibodies or IgG antibodies, which is depending on the infection, on the time point of the infection. And what is also necessary to know is about the vaccinated or non-vaccinated in the horses, which you can either do by anamnesis or there is also a possibility in some test systems to get some hints about vaccination or non-vaccination. And of course, it's necessary to have some multi-species format because we want to test the horses and the poultry. This kind of setup system is used to especially look in serum for the presence of West Nile virus antibodies. And if there is some material, of course, we perform PCR, especially to differentiate between the lineages. If we uh, get all this together, then we see those data from the last years, starting from 2010. So what we observe, or what we observed was 2024 was a very high West Nile virus year with a lot of humans, uh, compared to the other years, but also the horses and the birds were found positive. This year, so far, we have uh, just two birds and one horse. So, kindly speaking, it's a little bit a boring year for West Nile virus. Um, we the reason, therefore, we can maybe discuss later on. But during the last year, we also encouraged horsekeeper to use the vaccine. So that could be one of those yeah parameters which dropped the data. What is also very interesting is when we observe the starting point, the occurrence of of the West Nile virus in the year, then what we observe is that in the year 2024, the West Nile virus season started earlier, which um, is I mean, it seems to be an indication, uh, maybe for some climatic relevant things. So this is what kind of intriguing finding which we will observe in future. When we combine all the data, as I said, from animals and humans. Unfortunately, this map is available in German, but the darker the golden color is, the more humans were infected. The horses are the circles and the birds are the rectangles. And what we can see is this overlap, especially in 2024, in the eastern part of Austria, where human cases and animal cases uh, can be found in almost the same area. When we have a closer look over the years, then it's combining the data from 2016 till now. We still can see this kind of clustering in the eastern part of Austria. But we do also have some cases in the rest of Austria. What's also obvious here is this the purple bar rectangles and circles indicate clearly that we have 2024 as a very heavy West Nile virus year. We also had some interesting finding of an alpaca in Styria, and more recently, a very interesting finding in Tyrol, which is kind of really intriguing. It was a flamingo which hit the high voltage line and came to death. And thanks to the finders, it was sent for investigation. And interestingly, this is lineage 1. Whereas all the other samples, all the other data I presented you were lineage 2. So this is kind of very interesting finding. It's also, as far as I know, it's the ninth sighting of a flamingo, and it seems to be separated from some group which was either coming from France or from Italy. And there are some ideas that maybe due to the West Nile virus infection, there might be also some confusion that this poor guy just maybe missed his group or go to the wrong direction. So, but this is all, of course, assumptions, and it's a very rare finding and an interesting finding. Coming to what we can do, actually, for birds, horses, and humans, there are no therapies. So this is also many cases are asymptomatic, but those which have central nervous system symptomatic uh, can be very bad and can lead to fatal outcomes. There are vaccines for horses available, which as which could be the case that if you use them to drop the infections in the horses. And as far as I know, something similar happened in the US when there was a kind of introduction of the vaccine. Then the cases in the horses dropped. So this could be a good way to protect at least the horses. And what else you can do is to have prevent yourself, the horses, the birds, if you have some captive birds, from mosquito bites. So either using nets or maybe also management tools to bring the horses outside when there are less mosquitoes and so on and so forth. But this will bring me uh, to my colleague who will present you about the mosquitoes.
Okay, perfect. Then I want to say thank you to the LBE for the great introduction, and thank you, Georg, for your great first presentation. And now we will go from the veterinary side of the West Nile virus to talk about the vectors of the West Nile virus, the mosquitoes. I'm Barbara Seebacher. I work at Aedes for the Department of Vector Borne Diseases, and I'm involved in the first nationwide surveillance of West Nile virus in mosquitoes. What can you expect from my presentation? You can expect a general overview on West Nile virus and its close connection to mosquitoes, as well as relevant information about the mosquito biology. Afterwards, I will tell you about Austria's first nationwide West Nile virus surveillance project, One Health SURVector. I will tell you about our aim, what we are doing exactly, our results we have so far, and the challenges we faced so far. So mosquitoes are small, annoying, sometimes they don't let us sleep, and they are in fact world's deadliest animals for humans. They cause around 1 million human deaths a year. They can be vectors for parasites as well as for viruses. Regarding parasites, malaria is probably the best known disease caused by the parasites from the genus Plasmodium. But for example, they can also transmit parasites from the genus Staphylococcus. When it comes to viruses, mosquitoes can transmit a wide range of viruses like dengue virus, Zika virus, chikungunya virus, West Nile virus, and many more. Important to know is that not every mosquito can transmit every virus or every parasite. The capacity to transmit the virus or parasites varies greatly between the mosquito species. For example, only mosquitoes of the genus Anopheles can transmit parasites that cause malaria in humans. The feeding pattern, the host preference, and the virus replication rate are all key factors in the vector capacity across the mosquito species. But generally speaking, mosquitoes are great vectors. The female mosquitoes require blood meals to lay their eggs. They can take multiple blood meals from multiple hosts. They're highly adaptable. They have a rapid reproduction and therefore show up in high numbers. One Culex mosquito lays around 50 to 200 eggs with each oviposition. Here you can see the life cycle of a Culex mosquito. After mating, the female goes on to find a calm and steady water surface. This can be any water surface like puddles, lakes, rain barrels, or also water and plant sources. So it can be natural water surfaces or also artificial water surfaces near humans. The female goes on to lay 50 to 200 eggs in an egg raft, which swims on the water surface. From this egg raft, the larva hatches after 1 to 2 days, and then the larva feeds in the water on detritus and goes through 4 stages. The first stage, larva, doesn't feed anymore and finally molds into a pupa. From this pupa, the adult mosquito emerges. The development time from egg to adult mosquito takes about 10 days to 4 weeks, depending on the water temperature. Each round of reproduction for a female mosquito from blood feeding to egg laying is called a gonotrophic cycle, and a female goes through more than 1 gonotrophic cycles usually in its lifetime. The length of this cycle is influenced by factors such as temperature or the availability of hosts. When a mosquito takes a blood meal from an infected bird, the host, the virus goes into its digestion system and then multiplies in the mosquito until the virus reaches the salivary glands of the mosquito, and then from there they go into the hosts when the female takes a blood meal. This can be birds again, or also accidental hosts like humans or horses. And this transmission is called the horizontal transmission. In addition, it is possible that the female transmits the virus to its offspring, the eggs, while egg laying. So when these eggs hatch and develop into the adult mosquito, we have adult mosquitoes that already carry the West Nile virus in them without they even had a blood meal. This is called a vertical transmission. Here you can see a list of the possible West Nile virus vectors in Europe. While there are many mosquito species that can transmit West Nile virus, the vector capacity is the greatest in the Culex mosquitoes, and especially in Culex pipiens, Culex terentium, and Culex modestus. Culex pipiens, the so-called house mosquito, is a species which includes different ecotypes that can also, under specific circumstances, produce hybrids. We have the Culex pipiens form pipiens, and the female of this ecotype feed mainly on birds. They feed outdoors, they rest outdoors, and they need a blood meal to lay the first batch of eggs. The Culex pipiens form molestus. These females feed mainly on humans and other mammals, and they rest indoors. They can lay the first batch of eggs without having to take a blood meal, and they don't go into winter hibernation. So this makes the hybrid forms between these two ecotypes especially important to explain the spillover from West Nile virus from birds to humans, because they feed on birds as well as on humans. What is a key factor for the vector capacity of the mosquito is the temperature of the environment. The temperatures influences the survival time of the mosquito because it enhances the mosquito season in the summertime. You might have noticed that this year in Austria, the mosquito season started unusually late because it was such a cold spring. While in other years, the mosquito season started much earlier. The temperature enhances the number of the gonotrophic cycles the female goes through, taking the female taking more blood meals and also laying eggs more often. The higher temperature shortens the development time from eggs to adults. For example, if the water has a temperature of 30 degrees Celsius, it takes around 1 week from egg to adult mosquito. But if the water has a temperature around 15 degrees, it is about 21 to 24 days. And it influences the so-called extrinsic incubation period. This is the period from when a mosquito takes an infected blood meal to when it becomes capable to transmitting the virus itself via its saliva. This extrinsic incubation period differs vastly between the species, but for example, for Culex tarsalis, the extrinsic incubation period is 1 week at 30 degrees Celsius temperature, while it is 3 weeks at 20 degrees. Here you can see our suspect Culex pipiens on the bottom right side. You can see a female in the process of egg laying. Here you can see the egg raft. It consists of all of these small single eggs. They stick together in this raft and this floats on the water surface until the mosquito larvae hatch. On the top left side, you can see larvae of the Culex mosquito in the third or fourth stage. They float right under the water surface and are connected to it with its so-called siphon through which they breathe. And here in the middle, we can also see already one pupa that developed. So after this overview on the connection between the Western virus and the mosquitoes and their biology, I will go into the specifics of our Austrian-wide Western virus surveillance in mosquitoes. And here you can see a few impressions from the fieldwork, one impression from the lab work, and some pictures of the mosquitoes, how they look to us under the microscope when we determine the species according to the morphological features of the mosquito. One Health SURVector is comprised of a consortium of 8 partner institutions across 5 EU member states, with Austria serving as the project lead. The participating countries are Austria, Czech Republic, Greece, Hungary, and Slovakia. All members are actively engaged in tick and mosquito monitoring, and our pathogen screening focuses primarily on Western virus and Osuto virus in mosquitoes. In ticks, it focuses on Borrelia, Crimean Congo hemorrhagic fever, and tick-borne encephalitis. The priorities in the vectors and pathogens differ among the different countries. Our aim regarding the mosquito surveillance in Austria is to implement the first nationwide harmonized and uniform Western virus surveillance in mosquitoes. We want to act as an early warning system, detecting the circulation of the virus in mosquitoes before outbreaks in other animals like horses or humans appear. We aim for an ideal sampling strategy with a risk-based selection of the trapping sites, and we want the results to be publicly accessible. The system can be easily adapted to other vector-borne pathogens. The project runtime is from 2024 to 2026. And then we have the project RACE. In this project, we keep on detecting the Western virus and also other vector-borne pathogens like dengue or Synpis virus. On this map of Austria, you can see our trapping sites. Each point is a mosquito trap. Sorry. We have 86 traps distributed across Austria. We made sure that there is at least one trap in each nuts-free unit. Nuts-free units are used to classify small regions across the municipal level. We put more traps in areas with previous human cases, and due to our risk-based approach, in areas with higher residential numbers. As you can see, there are generally bit more traps here in the east. Vienna, more previous human cases, also here around Linz. And since this year, we also have a few trap sites near birds, for example, at the research station or a bird of prey rescue center. For trapping, we use the BG Sentinel traps to lure the mosquitoes in. The mosquitoes are lured in with the smell of CO2. CO2 is what attracts mosquitoes to our breath. And here we produce the CO2 either with a mixture of yeast and sugar in such plastic bags, or we use CO2 from gas bottles. In addition, we use a chemical lure that mimics the smell of sweat. And once the mosquito flies to the opening of the trap here, it gets sucked in by a fan, and then the mosquito is sucked into the net. The net then gets extracted by our trap operator. The net gets into a box. The box goes into the freezer that kills the mosquitoes, and then the box is shipped to us. The traps are set up twice a month for 24 hours from May to October. So who does the trapping? The trap operators are organized by the public health authorities of the federal states. They are a very diverse group of people with mostly no previous experience in mosquito trapping. They can be employees of the public health authorities, they can be employees of educational institutions, or employees of mosquito regulation organizations. Also, we have some interested colleagues from Aedes and private citizens who act as trap operators. And I want to use this chance to also say, if you're interested in operating a trap yourself and contributing to the project, please contact me afterwards, and I will be happy to give you further information about it. So after the frozen mosquitoes arrive at our lab, first we determine the species with their morphological features under the microscope. Then we pool the mosquitoes together based on their species and sex in Eppendorf tubes, up to 10 individuals a tube. Then we perform the pathogen detection with PCR, and if needed, we also do a sequencing. Here you can see this really nice infographic with the results of last year, 2024. We caught 7,927 mosquitoes, which are about as high as the Viennese giant Ferris wheel, in 1,364 pools. 3 of these pools were positive for West Nile virus, 2 pools in Burgenland and 1 pool in Styria, Steiermark. And when we look at the species composition of these mosquitoes, we can see that about 3/4 are from the genus Culex, which is very good. We don't differentiate here between Culex pipiens and Culex torrentium, because these species are so closely related that we can't differentiate them with their morphological features. But we do, with a specific percentage of them, we do PCR testing. This is again 2024. Here, positive mosquito pool in Styria, and here positive mosquito pool in Burgenland. The positive pool in Burgenland fits perfectly into all the other West Nile virus detection in humans and horses. Interesting is the pool in Styria, because this is the only West Nile virus detection we have there. Just the mosquitoes, no humans, no horses. If you look over the course of the year, we can see that the mosquito peak was in the beginning of July, and then it started rapidly to fall from the beginning of September. These are our preliminary results for 2025. Preliminary because the last trapping week is currently happening, and we are also waiting for some results from the sequencing. We can see that so far we have 5,817 mosquitoes, analyzed in 1,166 pools. When it comes to species composition, we have again around 3/4 from the genus Culex and our target species, Culex pipiens, Culex torrentium. Over the course of the year, we see that the mosquito peak was this year much later in the season. This was the beginning of August, because this was a period where it was warm and before there was rain, which is perfect for the development of the mosquitoes. In springtime, it was unusually cold, so the mosquitoes weren't able to develop as good. And overall, we have 20 to 25% less mosquitoes than last year. So we have this super empty West Nile virus detection map from 2025. And the combination of much less West Nile virus vectors with a higher vaccination rate in horses can be one explanation for this much more empty map. And last but not least, let's dive into some of the challenges that arose so far. So the first one is catching the West Nile virus and catching the mosquitoes. We need more mosquitoes to detect the West Nile virus. Our catch rates are too low. And to tackle this, we tried to adjust the trap sites this year. We're putting more traps into more rural areas where there are more mosquitoes usually. We tested if CO2 from yeast and sugar mixture or CO2 from CO2 bottles attracts more mosquitoes, if there is a difference or not. And we want to test another trap type last year, the gravity traps. So far, our goal of having an early warning system with the mosquitoes is not quite achieved due to the low trapping numbers. And also the repeat processing needs a lot of lab and personal resources to process all the samples fast enough. The next big challenge we face is to catch enough trap operators. Gladly, we work together with the public health authorities, and still it's not so easy finding volunteers. The traps have to be set up regularly over the course of 6 months a year. And a lot of our trap operators stay with us for 1 year or 2 years, and then they can't go on due to personal reasons. So it is always a challenge to find enough new volunteers. And we are still in the process of catching future prospects. So this was it from me. A big thank you goes out to all of the SURVector team, especially my colleagues Karin, Julia, and Severin. Big thank you to all the public health authorities that take part in this project, and of course the trap operators. And of course, a big thank you to you and your attention. And I'm open for questions. Thank you.
Okay, thank you very much, Georg and Barbara, for these